So the next topic I want to go over is glycolysis. This is where we're going to begin actual metabolism of carbohydrates, how our body uses carbohydrates to get energy. Glycolysis is the first step, and it's also the step that every living organism uses. It's a basic energy producing pathway that all organisms use, from bacteria, plants, humans, everybody. It's highly inefficient compared to aerobic respiration, which is what we mainly use as humans, but it is faster and it doesn't require oxygen. So sometimes, for example, in our muscles, when you're working your muscles really hard, they'll actually switch to using mainly glycolysis, and there are bacteria and other organisms that don't use oxygen. They don't use aerobic respiration, and they only rely on glycolysis. And there is another more advanced form of glycolysis that they use, but we don't have to go over that because it's not directly tested on the MCAT. MCAT really only tests on glycolysis and the human next steps, or actually the animal next steps. This process involves the breakdown of glucose into pyruvate, and it results in 2 molecules of ATP and 2 molecules of NADH. NADH is a very important molecule in ATP production, which we'll cover next week. Our body directly can convert NADH into ATP through a process called the electron transport chain and ATP synthase. But like I mentioned, we'll cover that next week. And 1 molecule of NADH is equivalent to about 2.5 ATP each. It's a little bit up for debate exactly how much, 2.5, but the more modern literature says 2.5, so that's what I'm going with, and I think that's what the MCAT mostly uses, but I think they'll mention it if they ever want you to count. But yeah, 2 molecules of ATP and 2 molecules of NADH per glucose. And like I mentioned, and if you remember from the digestive system lecture we did last week, if you have a complex carbohydrate, our body, before it even gets to this stage, it'll break it down into glucose, into a very simple sugar that it can digest, glucose, and then it'll be given out to the cells to use. So you wouldn't have something like sucrose going into your cells really, because our body can't really break that down into energy. It's kind of useless. Okay, so in terms of, and this can apply to glycolysis, pyruvate dehydrogenase, and the Krebs cycle, which I'm going to cover today. The steps, I think you should know the general steps and some of the names of the molecules. For biochemistry class, you may have had to memorize the exact mechanism and any enzymes that were used. For the MCAT, you don't really have to know that. It's extremely unlikely that they'll ask you about an enzyme or something like very, very specific about the process of a specific step, just because it's way too in detail and way too much for you to memorize. But you should know the general steps, what the logic is behind them, what molecule starts and ends, which steps produce ATP or NADH, right? Okay. But you'll see as I go through. So the first step is you phosphorylate glucose to glucose 6-phosphate. If you guys remember, phosphorylating something is kind of like, I want to think of it as activating something. By phosphorylating something, you activate it in most situations, right? So, and also in order to phosphorylate something, you actually need to get that phosphate from somewhere. That somewhere is usually ATP. So you actually take a phosphate from ATP and put it to work somewhere else, right? And this may seem counterproductive. Well, why, if you're trying to make ATP, why are you using ATP to activate? Well, it's because you actually need to use some of it to start the process, but you end up making more than you use. So it's actually a net gain, right? So this first step uses 1 molecule of ATP, but it's necessary to be in the process. Again, this is what glucose looks like in kind of like a linear format. And you just added this PO4, this phosphate, onto this carbon at the end, okay? To activate it to the 6th carbon. Next, we need to move the carbonyl group from C1 to C2. This turns the aldose glucose into the ketose fructose. The current molecule at this stage is fructose 6-phosphate. we'll see exactly like this needs to happen in order to progress, and we'll see in the next step why that is. Okay, just know that immediately you turn glucose into fructose. The next step is making the molecule more symmetric. Since there's this is no longer an aldose, we can actually phosphorylate the C1 carbon. That's why we needed to change it, because if you remember, aldehydes have a double bond O on the end, which cannot be phosphorylated. So we actually need to make it a ketose and move the double bond O from the carbon 1 to carbon 2, so we can actually phosphorylate the carbon 1. So, and this will make it more symmetric, and you can see here, this was originally already here from the first step. We just added another PO group right here onto the C1. You know this is the C1 because this is where the double bond O is, at the C2 carbon. Okay, and now you have 1,6-fructose biphosphate. Now that the products are somewhat symmetric, we can now cleave the 6-carbon fructose into 2 3-carbon glyceraldehyde phosphates, right? We can make the one really long 6-carbon chain into 2 smaller 3-carbon chains, each with their own phosphate group. Okay, so you have technically there is an intermediate. So when you do this, initially you get one of these, which is the glyceraldehyde phosphate that I mentioned here, the 3-carbon glyceraldehyde phosphate. You also get this, right? Because these are kind of symmetric, but they're not exactly symmetric, right? You can see that these are a little bit different because this has double bond O on the end, this one has in the middle, right? But this will, in the exact next step, this will get converted into this. So you'll actually have 2 of these now. Okay, so initially you get 2 slightly different molecules, but then one of them just gets immediately converted into the other one. So that's why I didn't bother with the intermediate. But I just think you guys should know that. Okay, so you have, in the end, you have 2 of these. Okay, so far we've had to phosphorylate twice, and we've actually had to use 2 ATPs already, right? So we're at a pretty significant net loss, right? There isn't another phosphorylation event which turns the glyceraldehyde phosphates into biphosphates by adding another PO4 to the opposite. So again, we're actually phosphorylating one more time for each 3-carbon, right? And since we have 2 3-carbons, we now have to use up basically 2 more phosphorylations. But these don't use ATP. Rather, these use NAD+ as a cofactor, and it actually turns it into NADH, which is what we want. We want to turn NAD+ into NADH. So rather than using energy using ATP, we actually make something NADH, which is equivalent to ATP. So this is a very net positive step. And again, this occurs twice. So 2 NADPH are produced, and NADH are produced. Again, because there's 2 of these, right? And each one gets an extra phosphate. Okay. This phosphate group is immediately removed and is used to convert ADP into ATP. So we put the phosphate on by using NAD+, and then we immediately took it off and put it on an ADP to make ATP. Okay. This is the actual direct energy making step. NADH it's technically energy, but it's not an energy making. It's not directly energy, whereas this is directly making an energy. It produces 2 ATP because you do it twice, one for each molecule. Lastly, the first phosphate group that we originally put on is removed, and it is again used to turn ADP into ATP. And again, this produces 2 more ATP. Ultimately, this produces 4 ATP and 2 NADH, but we had to use 2 ATP to actually for this process. So we have a net gain of 2 ATP, 2 NADH. Okay. I know there's a lot of like letters, but try and follow. And if not, please let me know in the chat if you want me to explain again. The final product of all of this is called pyruvate, right? And it's shown right here. This thing called pyruvate. Okay. Now, for a lot of organisms, this is, somebody has a question?
Mark, is it, will they ask us about committed steps since you mentioned that the MCAT doesn't go in detail?
Sorry, could you repeat that? Which kind of steps? I didn't hear that word.
Since you said the MCAT doesn't go in detail, would they be asking us about committed steps and stuff like that?
No, I don't think so.
Okay.
I don't think so.
All right.
Because that's that's under more, that's very specific, because that also involves knowledge of enzymes, and like maybe you can memorize what are the committed steps, but that's kind of useless if you don't know why they're committed steps, and in order to know why, you would have to know a lot about the specific enzymes, which would be too much for them to ask, I think.
Okay.
Yeah, so they don't really ask that. Okay, so this produces pyruvate, and for some organisms, this is kind of one of the end stages. It's not actually, but for the purpose of the MCAT, it's one of the end stages of this whole process for glycolysis. But for us humans and other animals and a lot of like plants and stuff, they'll use oxygen. We can actually continue, once we have this pyruvate, we can actually use this pyruvate and as fuel as well, and break it down even further and get a lot more energy out of it than we can just from converting glucose to pyruvate. Okay.